Hello students and welcome to Lecture 20. Today we're going to dive deeper into the apoptosis mechanism. But before I begin, I want to just remind you of a little bit of context, as we always like to do. So we are, of course, in the third unit of our course thinking about protein pathways. And we're thinking specifically about the way that ligands interacting with receptors can tap into an intracellular network of proteins that do signaling. So we'll represent it kind of like this. And we've covered the classes of ligands, we've covered the classes of receptors, and we've covered the major signaling pathways. Recently, we looked at a new type of ligand, nerve growth factor, and remember, this ligand is representative of the survival factors that influence cell life and death. And specifically, ligands like NGF suppress apoptosis. Remember that apoptosis is that regulated form of cell death, that neighbor-friendly form that we talked about, that's quite normal in developmental context or throughout the life of an organism, if we're talking about turnover of cells or development of the system, but it's very abnormal in disease states like Alzheimer's. We learned how NGF signals through the receptor enzyme called TrkA, and remember that's the receptor tyrosine kinase that NGF binds to with high affinity, and that this induces a signaling cascade. It actually induces sort of parallel divergent cascades. And we heard about how phospholipase C pathway can be upregulated, and that's really the PIP2 pathway. We heard about the RAS, ERK, MAP kinase pathway. Remember, that's the one that influences the outgrowth of the axons, make them really grow much further and more robustly than normal. And most importantly, we talked a little bit about this PIP3 pathway. That's the one that involves PI3 kinase, PDK, AKT, and bad phosphorylation. And remember that that leads to survival. And of course, another way to represent this is it inhibits apoptosis. So the survival pathway of NGF is the one, obviously, that regulates and controls apoptosis. Okay, so that's the context. Where does today's lecture fit into all of that? Well, today we're going to focus on a lot of this and the role of BAD and phosphorylated BAD in promoting cell survival and inhibiting apoptosis. So we're really diving into the apoptosis mechanism today. And in order to do that, we're going to introduce a new family of proteins called the BCL2 family. BAD is a member of the BCL2 family. And we'll see that BAD and other members of this family are central to the apoptosis mechanism. Okay, so that's what we're all about today. All right, last little bit of context, because on Monday we'll be hearing Lecture 21. Lecture 21 is going to be another one of our primary articles. And remember, a primary article is a data-driven scientific article. We covered one earlier in the class when we looked at the cloning of the beta secretase. I'm going to cover another one with you, and that's going to be looking at the paper that first documented AKT phosphorylation of BAD. And what that's going to do for us, it's going to be another one of those kind of bottle episode-like lectures. It's going to let us really dive deep into this regulation of the BCL2 family via this PI3 kinase pathway and growth factors. And it's going to be another sort of introduction to you of the primary literature. So it'll serve kind of both of those purposes. And that's kind of that's what we'll be doing today and then again on Monday. All right, let's look at the outline. So here's my outline for today. And basically I've cut it in half here. This part is part 1, this part is part 2 of Lecture 20. And I'm going to begin with a sort of reminder of the suppression of apoptosis pathway that growth factors can induce. And then we'll place the BCL2 family within that pathway. We'll look at the mechanism. And I'll do a little bit of board work or whiteboard work here, as well as walking you through my PowerPoint slides. And that should make it really relatively clear. Part two, we'll look at how well conserved these pathways are evolutionarily. And then we'll look at some other aspects of the downstream mechanism. Turns out it involves a class of proteases. So isn't that a nice way to come full circle in this class? We're going to end up looking again at proteolysis as an important regulator. And here the proteases are called caspases instead of secretases, for example. All right, let's dive in. All right, so this slide does a good job of kind of giving us an overview of how survival factor signaling through receptor tyrosine kinases will induce this pathway that activates AKT and then allows for bad phosphorylation. Let me walk you through it. So remember, the survival factor, we think of this, you can just think of this as nerve growth factor if you like, but it could be any of the other similar types of peptide signaling ligands. Let's imagine that it's out there in the extracellular space, and here's our receiving cell. The cytosol is here, the extracellular space is here. The NGF ligand allows the receptor tyrosine kinase, this would be TRK-A, to dimerize. And that activates, and we get the cross-phosphorylation deal. We get the phosphorylated tyrosine, okay? So now that we have the active receptor enzyme, SH2 domain containing proteins can be recruited up to the membrane and bind to that phosphorylated tyrosine. So phosphatidylinositol 3 kinase, PI3 kinase, is an SH2 domain containing protein, and it can interact with the activated receptor enzyme TRK-A. And what's cool about this is this brings this protein out of the cytosol and up to the membrane, where now it's in proximity to its PIP2 substrate. So now we have kinase, and it's activated in proximity to substrate, and the phosphorylation converts PIP2 into PIP3. And now remember, it's not just one PIP3 that's made. There's 1, 2, 3 shown in this cartoon, but there's obviously hundreds in this local space of membrane. So that then allows other proteins to be recruited up to the membrane. So now PDK, which is a kinase, and AKT, which is a substrate, are recruited. And now they are close to each other. And PDK is going to phosphorylate AKT, and here is AKT, once phosphorylated, moving away from the membrane, and now moving through the cytosol to act on various things, various substrates. And we're going to ignore these other ones, and we're only going to focus on this one because that's the one that we are going to care most about. So AKT, when activated, can phosphorylate BAD. And remember, when phosphorylated, this promotes cell survival. Or another way to represent this would be this inhibits apoptosis. All right, so that's the context for the survival factor signaling and how it will start to prevent apoptosis. But how? How is it that phosphorylated BAD leads to cell survival and suppresses this intrinsic apoptosis pathway? It turns out that BAD is a critical regulator kind of in the middle of the cell death pathway, okay? So before we get into the nitty-gritty molecular details, let me quickly introduce the family of proteins that BAD belongs to. That's what we call the BCL2 family. Here's a look at the BCL2 family proteins, and the first thing that we need to know about this family, like many families, mine very much included, there are a lot of members and there are very different personalities and different roles. So we can kind of break the BCL2 family into what we might consider the good members of the family and the bad members of the family. And I put those in quotes because obviously I'm, being colloquial and I'm being analogous here. Proteins are neither good nor bad, they're just proteins, right? But from a cell survival perspective, the good members promote cell survival and the bad members promote cell death. So that's kind of interesting, a dichotomous family with, good guys and bad guys, so to speak. So on the good side of the ledger, we have proteins like BCL2. Yes, the BCL2 family has a member named BCL2, not just to confuse you, but just for historical reasons. This was the first member of the family discovered, so the whole family got named after that member. BCL2 is a good protein from the perspective of it being anti-apoptotic. And of course, that means pro-survival. So BCL2. BCL2 has multiple of these domains that are indicated here in the cartoon, and those help it to perform a good function, a suppression of apoptosis function. And there's other members of the family. There's another one that's pretty popular, BCLXL is one. And what's interesting about these good members of the family is they can homodimerize. To homodimerize means to bind together, so two of the same, that's where the homo comes from, dimer, two. So two BCL2 proteins can dimerize like this, and two BCLXL proteins can dimerize like this, okay? All right. Now, what about the bad side of the column? Well, there are multiple members of this family who are pro-apoptotic or anti-cell survival. I'm not going to list all of them here. I'm just going to list a couple for you to be familiar with. One of them is our protein BAD that we mentioned before, and that's appropriate. The BAD protein is bad from a life or death perspective because it promotes the killing of a cell. Another one that is on the bad column is BAX. Okay. And like BCL2 and BCLXL, BAX is also capable of homodimerization. Okay. And kind of another neat thing about this protein is not only can we have homodimerization within a column, we can also have heterodimerization between the columns. So that means two different subunits bound together. Like, for example, BCL2 can heterodimerize with BAX. I'll just represent it like that. Okay. So what is all, and by the way, BAX can also homodimerize. If I didn't mention that before, maybe I did. Anyway, you get the idea. Homodimerization, heterodimerization, etc. So that's the BCL2 family. Okay. So this business I'm going to talk to you about on the next slide, and I'm just going to pull up a little whiteboard here and do some free drawing because I think it's going to make it a lot more clear. All right, so how does all this work, and how does the BCL2 family help to regulate cell death? In order to take a look at this, I'm going to kind of represent a cell like this. Here's the membrane, and down here is a mitochondrion. It turns out that mitochondrion are very important organelles in regulating cell death. I bet you didn't know that. It's kind of an interesting thing about cell biology. So up here in the membrane, we have our receptor and our ligand, and we induce this cascade that I'm just going to represent with these arrows. We get AKT activated, and then we get phosphorylation of BAD. And we know that this is somehow promoting cell survival by inhibiting apoptosis. So how does it all work? What I'm going to do is I'm going to represent remember the mitochondrion has a dual membrane system. In the outer membrane of the mitochondrion, that's where a lot of the BCL2 family members reside, including BCL2 itself, which I'm going to represent as this oval, and Bax, which I'm going to represent as a kind of rectangle slash square. Okay, so these proteins reside in the outer membrane of the mitochondrion. They're kind of anchored in with a little lipid group, and they stick out into the cytosol. And what we want is this situation where BCL2 is heterodimerized with Bax, like so. The reason we want that is that this is pro-survival. This heterodimerization is pro-survival, okay? And as long as BAD is phosphorylated in a growth factor dependent manner, this is what is going to predominate in the cell, okay? But what would happen if we didn't have the growth factor and we didn't have active AKT and we no longer had BAD phosphorylation? Well, now, and I'm just going to represent BAD as a triangle. Now BAD is not phosphorylated, okay? And it's going to diffuse over here, and it's going to grab onto BCL2. So I'm going to draw that little triangle, try to draw it like that. What that does is it pulls the BCL2 protein away from Bax. So we end up with this situation where the oval-shaped BCL2 heterodimerizes with unphosphorylated BAD. Okay. And of course, what that means is that now Bax is no longer bound by BCL2. Okay. What happens when Bax is no longer bound by BCL2 is Bax can homodimerize with another Bax subunit. And this is really problematic for the cell because it punches a big hole in the mitochondrion membrane. And what happens is it forms a little pore that a molecule called cytochrome C can escape through. You may have heard of cytochrome C before. It's a member of the electron transport chain. It helps to ferry electrons from one complex to another during respiration. Well, once we have a Bax homodimer that has punched a hole in the membrane, we now have a pore through which cytochrome C can escape the mitochondrion. And it ends up here in the cytosol. Once that occurs, we are irreversibly committed to apoptosis. And the reason for that is cytochrome C in the cytosol induces a multi-step cascade that leads to the activation of those proteases that allow for the execution of the cell via the apoptosis mechanism. So you don't want cytochrome C in the cytosol, you want it sequestered in the mitochondrion, and the way we do that is we prevent Bax homodimers from forming. And of course, the way we prevent Bax homodimers from forming is to sequester or trap Bax with BCL2. But we can't do that if BAD is in the unphosphorylated state, because unphosphorylated BAD heterodimerizes with BCL2 with greater affinity than BCL2 binds to Bax. So, in other words, BCL2 would rather be with unphosphorylated BAD than with Bax. So the whole thing needs to be, this whole thing can be regulated by the presence of that growth factor, the activation of AKT, and then the phosphorylation of BAD. Because when BAD is phosphorylated, guess what? It loses its affinity for BCL2. And that's how we suppress apoptosis. It's really an amazing mechanism. Here's a look in a much more attractive form at what I just showed. And first thing I'm going to do is I'm going to just kind of break off this part of the diagram and just kind of focus on this part here. So it's really about the same thing that I showed. It's not quite as involved upstream. But look, here's the mitochondrion. Here are the proteins within it. So in red, we have Bax. In looks like kind of purple to me, we have BCL2. And then here represented we have bad. It's more like a teardrop here than a triangle, but you get the idea. And this would represent unphosphorylated bad. When unphosphorylated, bad can come down here to this heterodimer of BCl2 and Bax and heterodimerize with BCl2. And that's what that is showing you right there. So now we've pulled the BCl2 Bax heterodimer, we've pulled the BCl2 away. And that leaves Bax available to do this, to homodimerize. Occasionally, Bax will be further bound to unphosphorylated bad. Don't worry too much about that detail. Just know that homodimerized Bax is where we get the pore in the membrane, and that's what allows the cytochrome C to escape. And once that cytochrome C escapes, what it does is it occupies some binding sites on this protein here called APAF1. And when that happens, we have activation of this enzyme called a caspase. A caspase is the protease that executes the cell. And indeed, we call these, caspase 9 is what we call an upstream caspase. Caspases 3, 6, and 7 are downstream caspases or executioner caspases, because they do all the things like fragmenting the DNA by activating a nuclease, breaking up the chromatin, breaking up the cytoskeleton and other surface features of the cell. They basically digest the cell from the inside. And remember, because in apoptosis the membrane is never disrupted, all of this is going on inside an apoptosing cell, and it doesn't spread in the tissue. So cytochrome C release from mitochondria via Bax homodimers activates a cascade that gives us this protease activation that then allows for the execution of the cell. That's the idea. Okay, here's my last slide for part 1 of lecture 20. And I just wanted to kind of formalize some of the ideas with a little different schematic, but I think it's helpful. It helps us to think about how all this works. So we're looking at 2 different cellular states here in this cartoon. Over here is the survival side, and over here is the apoptosis side. So let's see what's different about them. First, the survival side. So here with NGF present, here's the NGF ligand, that's a survival factor. We get the dimerization of TrkA, and we're not showing the detail of the activation, nor are we unpacking all of the upstream signaling components. We're just representing them with that arrow, okay? And because that arrow has an arrowhead, that's an activating arrow. That means it's going to stimulate whatever is next, okay? And what it's showing us is it stimulates the protective role of the BCL2 family. And what that does is it inhibits APAF1. And look, it's totally schematic, right? We know there's a lot of mechanistic detail in between these 2 steps. The whole thing of homo and heterodimerization, cytochrome C release, all of it, all that's represented just with that one little blunted arrow to show that if we activate BCL2, we suppress APAF1. And that means that caspase enzymes remain inactive, and we end up with cell survival. Okay? Over here, what's going on and what's different? Well, now we've taken the survival ligand away. And now there's no way to activate the protective role of BCL2. So this inhibition that we saw over here is gone. It's out of the way. That means APAF1 is disinhibited. And now it is able to stimulate the cleavage and activation of the proteases that execute the cell death pathway. And remember, what's really making all this happen is the homo and heterodimerization of the BCL2 family, as we talked about in the earlier slide. So this gives us a nice schematic way of looking at these signaling pathway, and there's another couple cool features here. For example, here's the ATP dependence, and part of the reason that ATP is necessary for apoptosis is APAF1 is in part regulated by ATP. Of course, another reason is there's a lot of phosphorylation going on in these cascades. And so that's another reason that we need a lot of ATP around, in addition to driving the metabolic pumps that keep osmoregulation at normal levels. Okay, so that's the end of part 1 of lecture 20. I'll be back after a short break for part 2. And what we're going to do in part 2 is take a look at how well conserved these pathways are, unpack the pathway just a little bit more, and spend more time on this downstream part of the pathway. Okay, back after a little break. See you then.